So, welcome to Human Physiology Bio 224 Laboratory. Laboratory is going to have kind of two main focuses during the semester, and I wrote it up here. One is going to be demonstrations of lab concepts. There's going to be more just showing the basic topics that we've been talking about in lecture and showing them more in a hands-on way so you can get a better feel for them. In these demonstration labs, you'll be doing experiments, but it's going to be more about just recording your observations and then interpreting and putting in context what those observations mean. The other big part of our lab, though, is going to be about learning more about the whole process of science. You have your lab textbook, or actually your class textbook, and it tells you all this information about the human body, but how do we know? Where does that knowledge come from? And there is this long-standing process, the scientific method that we use to ask questions and find answers and get our best possible idea of what is going on here. So that's going to be a big part of our lab as well. And these are going to be these hypothesis-driven labs where we're asking questions. That's like the objective is actually just what is this relationship between one variable and another variable? Like, how does the amount of coffee you drink relate to how well you end up doing on exams? Or, we'll talk so much more about independent and dependent variables as the semester continues. These will also include hypotheses, where you have to kind of think about, well, what do you think the answer to this question is, before you do the experiment. This does not have to be right. In fact, if you knew the answer, you wouldn't have to do the experiment. People often get kind of uncomfortable getting into this zone because they don't like to be wrong. But it's fine to be wrong as long as your guess, kind of makes sense at some level, and you have like a why, like a reason why that makes sense. You don't want to just like pull something out of the air and say, "There's my guess." That would not be all right. But the hypothesis very well might be wrong. That's part of science. That's actually really good information for other people to know, like, all right, that's not the way I should go. Let me go look at some other place. Sources of variability is a really important part of this process as well. Whenever you're doing experiments, there's all sorts of things that can affect your data that could actually really make the whole experiment useless. Like, we're going to be doing a jumping lab, looking at how high people can jump and how different body measurements affect your height of your jump. And again, in the lab later, we'll talk about sources of variability, but if somebody's wearing Birkenstocks, or someone's wearing some crazy basketball shoes, or somebody is got horrible body arthritis and can barely move, and somebody is like a trained athlete, those are going to really affect how high you can jump. Those are sources of variability. You've got to think about that before you actually do the experiment, so you can make sure you design it in such a way to minimize the effect of variables that you are not interested in. So, in fact, I can, for the labs that are demonstration labs, and in your schedule, there's actually a little mark on the ones that are hypothesis-driven versus the ones that are not. If it's a demonstration lab, you have to have in your lab notebook the description of the lab. You have to have the methods. You have to record your data. You have to have a discussion of what the data means. But when you get to the hypothesis-driven labs, there's another layer that you have to worry about, and attend to, and that is really be clear about what is the question you're asking. Why are we doing this? This is more than just demonstrating concepts. There's a question, an objective. The objective, and it's always going to be a very simple, straightforward question. What is the relationship between the independent variable and the dependent variable? And again, we're going to be going so much more into independent and dependent variables. If your objective is not a simple, straightforward question between these two variables, then you've got to kind of go back and write it in a very, I like to use the word concise. The more concise, clear, and simple you can be with what the question is you're asking, the more able you are to design a nice, clean experiment to answer your question. The hypothesis, which is, again, the objective question we are trying to answer. The hypothesis, what you expect to find, but it has to have a why. This could be helpful, like, if people drink more coffee, then they will perform better on their physiology exams. You gotta have the reason, because caffeine is a central nervous system stimulant and amps you up, and you're gonna be able to stay awake longer and study longer, and that studying longer is gonna make you do better on your exams or something. You've got to have a reason. You can't just say, more coffee, better scores on your exam. You've got to say, more coffee, better scores on your exam, because, and again, you don't have to be right. The hypothesis is a guess, but it has to be, we talk about an educated guess. You can't just kind of, pull it out of the air. And then sources of variability, you want to make sure you list those explicitly. Because if you don't think about them before you do the experiment, they might completely invalidate. You get all your data for this jumping lab, but you never actually made sure what kind of shoes people are wearing. There's a good chance that the shoes made more of a difference than any of the other things that you were measuring, so your whole experiment is for naught. So, lab book. So, you are going to be using this lab manual, and this lab manual has basically descriptions with the methods and materials for each of the labs we're going to be doing. Also includes like suggestions to help develop your discussions. It also includes some other stuff in here as well, a study guide. In the Appendix A, there is a study guide for the course you definitely are going to want to be referring to. But for right now, we're going to talk about the lab part of it. You'll need this. It's going to be used in conjunction with your lab notebook. So everybody needs one of these. And it has to be a very specific kind of lab notebook. It has to be bound. Notice it's not just a binder with loose leaf pages. Each page has a number on it. So the numbers are going to make sure that pages aren't ripped out or stuck in. This is going to be a true and honest record of what you did in the order you did it. You should have this graph paper, so it makes it easy to make tables and graphs in there. You have to write your lab notebook in pen. These are examples of lab notebooks. Pen, because this is true and honest. You can't go back and erase something. It's like, oh, I meant that. So if you are going to get rid of things, you just put lines through them. Let me see if I've got examples of that. I'll have to edit a little. Like right here, we have somebody who was changing her objective, and she just drew a line through and put a revised thing in there. It's not wrong to revise things. And I think this causes a lot of stress for students in this class because they're used to having this being judged on having the right answer. And this is a record of your work and of your thought process, and sometimes you change your thoughts. We will go over and review your objective or your hypothesis and say, you know what, you might want to, you didn't quite get what we're looking for here, and you might have to draw a line through it and write, here's my revised objective, my revised hypothesis. And some people are mortified to be exposed as not being perfect. And no, I'm not perfect. Tina behind the camera is not perfect. We all write things down and realize, wait a second, draw a line through and revise. So it's one of the things that I try to emphasize, is that this is not a perfect final presentation. You are going to be writing papers later in the class where we do care about your grammar or the formatting or this or that, but that's not this. This is a true, honest record of what you did in your lab. It will start in the first page. There will always be a table of contents. Again, this in your lab manual, it talks about how to put this out, but basically this will be a work in progress. Each time we do a lab, you have the name of the lab, the page that it is found in your lab notebook, the date that you are doing it. So this is also going to be used in your lab exam. The lab exams are actually open lab notebook. So you have a nice record of what you did. 2 months from now, you're going to wait, what did we do in that chemistry lab? But you can just go into your table of contents, all right, chemistry review, and it's on that page, and then assuming you did a good job of recording what you were doing, what you found, what it means in your discussion, this is going to be this amazing ally to help you totally ace your physiology lab exams. So you really do want to, you're going to get graded both on the lab notebook for itself, but doing well on it will also totally help you succeed on the lab exams as well. Other things about the lab notebook, you'll be pasting stuff in here. There's no reason to write the methods and materials, copy out of the lab manual into your lab notebook. You can use scissors, use paste, put things in here. You will also be putting in graphs. You can kind of see you have, in the graphs, whenever you put graphs in, you need to make sure that they have numbers. You don't want to just have orphan graphs. Anything that is pasted in here should be referred to in the discussion. So you can't just put a graph in here and then assume that somebody knows what it's about or why it's there. It should always have a number and be referred to in the text of the discussion. As we see in figure 1, there was no relationship between reaction time and the intensity of the light in the room, something like that. Also units. You never want to have numbers in your lab notebook that don't have units, right? The canary weighs 50. If it was 50 pounds, what the heck? Or 50 grams, that might make more sense, but 50 doesn't mean anything. So you need to always have units. Only exception, there's pure ratios like specific gravity and stuff, but it's almost always units. Make sure you always label the graph axes as well. Whenever you have a graph, you need to say what is the y-axis, what is the x-axis. In general, as a rule of thumb, the x-axis is almost always going to be your independent variable. As it changes, and then the y-axis is going to show what the dependent variable is doing as the independent variable is changing. Other things, part of that writing in pen, don't cross out, don't white out either. Also, don't just paste paper on top of things that you would rather we didn't know happened. It's, again, you are not graded on being some ideal human that never makes a mistake. You are being graded on, are you accurately recording the data, and ultimately, responsibly discussing and interpreting what it means. Like in your labs that are more about demonstrations, it's more, again, kind of interpreting just how do your observations fit into the concepts that we've been discussing in class. In the hypothesis-driven labs, it's going to be a little deeper. You're going to have to talk about how your data informs your original question, right? And your hypothesis. Is your hypothesis supported by the data or not? Or sometimes it's going to be a little ambiguous. Talk about what it means about the big question overall. Talk about how sources of variability might have played a role in influencing your results in a good or bad way. All of that will be part of your discussion in these hypothesis-driven labs. Like your discussion, first order business. You had your objective, your question, and your guess at the answer. What do you think the data has to say about that? And then you can go from there and continue to expand. So